Hello everyone, and thank you for coming to watch my presentation today. Today I will be talking about the Chytrid Batrachochytrium dendropatitis fungus, which is leading to global amphibian declines. My name is Kiana Johnson. I am a senior here at River Falls studying field biology and outdoor education with two emphases in Spanish and sustainability. And my future goal is to be an environmental educator, such as a teacher, a naturalist substitute teacher, or work at a nature center, work with the DNR, anything like that. I would just like to educate people on different aspects of the environment. So just a little overview today. I'm going to be starting with some background knowledge. Then I'll be talking about the Chytrid BD fungus, and then the disease that fungus can cause, called chytridiomycosis. And then just briefly at the end, I'm going to touch on just a little bit of current research that is going on within that scientific community. So to start off, I'm starting with just the definition and a visual of taxonomy, which is the science of classification. And so what this diagram is showing you are some of the different classifications that scientists can place animals, plants, anything like that into. And so if we start at the top, the domain and the kingdom are the broadest categories, where the genus down here and the species are the most specific. So like if we are talking about humans, Homo sapiens would be down here in the species and the genus area. So I just wanted to go through that real quick, just because I'll be talking about class, order, genus, and I might say species just a couple different times, and I just wanted to give you just a brief reminder of kind of what I'm talking about. So starting with class Amphibia, those are the amphibians, and that is comprised of 3 orders: Onura, which are the frogs and the toads; Caudata or Urodela, which are the newts and salamanders; and Gymnophiona or Apoda, which are the caecilians. And respective pictures are shown there. For the focus of this presentation, it's kind of general to amphibians, but for the sake of my examples and Chytrid BD in general, I'm focusing on Onura, which are the frogs. So why are frogs important, and who cares if they're here or not? So they play a variety of roles in food webs, starting with even their egg life stage. That's food for other animals, but even as they move throughout their life, they are predator and they are prey for different kinds of animals. So just in this food web right here, the arrows going into the frog represent that frog's prey. Arrows going out of that frog, that represents the frog's predators. In addition, they live in both the aquatic and the terrestrial environments, so they're susceptible to stressors from both areas, which makes them a good indicator species. And what an indicator species is a species that scientists can look at to kind of gauge the health of the environment that they live in. And so, say an indicator species population is happy and healthy, that gives good indication that that environment is also happy and healthy. But if an indicator species population levels start to decline, something changed within that environment, and that could be a detrimental change that could have a ripple effect among many animals. So it gives scientists a cue on changes that are happening. And lastly, I included a medicinal use infographic just because I think it's kind of cool, and I bet a lot of people don't know this about amphibians in general, but frogs as well. So if we look up here, that toad was actually used in the development of a high blood pressure medicine, or this frog was used in the development of blocking an HIV transmission, or over here, that frog was used in a diabetic foot ulcer cure. So just kind of some weird stuff, but frogs are important not only for the environment, but they're also showing more and more importance for human health as well. So in general, we're going to start looking at this map. Amphibians are widely distributed over the world. They stay out of the super cold regions like the polar regions because they don't have the food supply they would need to survive there, and it's too cold. But scientists are seeing global amphibian declines. So it's across the board, they're seeing it all over the world, and currently about 1/3 of all amphibian species are being threatened with extinction. And so scientists are very concerned, especially because they are indicator species. So if Like I said, if an indicator species population is declining, something is changing with the environment, and that is our first cue that we should investigate that and try and figure out what that change is. And so another thing that puzzled scientists is some of these disappearances were being seen in pristine areas, so areas that humans don't have much impact over. And so this was kind of concerning to the scientists because usually the causes of population declines are attributed to humans, whether that could be introducing invasive species, over-harvesting animals, altering the habitat, or destroying the habitat altogether. Humans could be leading to climate change. Humans could be polluting the environment, leading to contamination, or spreading infectious diseases. And so for the focus of this presentation, I'm going to be focusing on the infectious diseases portion. So finally getting into the chytrid fungus, what is it? So there's actually a family called Chytridium mycota, which are the chytrid fungi. There are hundreds of species within this family, and they mostly act as decomposers, focusing mostly on chitin and keratin. And chitin is that hard structure found in the exoskeleton of like arthropods such as insects or crustaceans or crayfish. And keratin is a protein that you can find in human hair and human nails. But for the focus of this presentation, I'm not generalizing to all chytrid fungi. I'm focusing on Chytrid BD. And so when news articles start appearing like this, a deadly salamander disease just got a lot scarier, or fungus turns frog into sexy zombies, or amphibian apocalypse caused by most destructive pathogen ever. Deadly fungal disease has devastated more than 500 amphibian species. This fungus has wiped out more species than any other disease. Or deadly frog fungus now thrives where we thought it couldn't survive. These articles started concerning the people more, and so more and more research and more funding got put towards the Chytrid BD fungus itself. And so why is Chytrid BD so detrimental? So I'm going to walk you through the life cycle of this fungus. It is asexual, which means it does not use sperm and egg to reproduce. It uses spores instead. So I'm going to walk you through this infographic over here, starting with this section. This represents an amphibian skin, so we're going to say a frog skin. Out here is the aquatic environment, so say a pond. In here is the interior of the amphibian. And so we're going to start with number 1 right here. This is a zoospore, so one of those spores, so that kind of shows you that it is an asexual form of replication. Once a zoospore finds the amphibian skin, shown right here, it's going to start to encyst and kind of make its way through that outer layer. Once it is inside, it's going to grow a bit. It's going to become a thallus. And then as that thallus continues its life cycle, it's going to progress into a zoosporangium. Inside of that, zoospores are going to start to be created. Here, you can see those zoospores. They're those circular discs. In addition, in the zoosporangium stage, this tube is created. This tube is called a discharge tube because the zoospores are going to be released through that tube in the following step. So what has to happen is this operculum, or this cap, is going to either become dissolved or kind of pop off, releasing those zoospores back into the environment. And so, as frogs and amphibians are getting infected by these zoospores, it can cause mass mortalities like the one seen in this picture. And so this is not just focused in, say, like the tropics, where there are lots and lots of amphibians. This is across the world. So these maps are showing areas where research is being done, and are showing, you can see up here in the key, if there are positive sites and negative sites, and then negative sites, but the sample size is unknown. So they don't know it actually could be there. It's kind of hard to find or get good samples to be able to run the qPCR to see if BD is present or not. And scientists wanted to figure out where this came from, so they started to do more and more research. So in 2002, 35 different samples were looked at. In 2009, 59 samples were looked at. In 2018, 234 samples were looked at. And this is the best study to date that I have been able to find. So it's a global collaboration between 38 different institutions. And their main conclusions from their paper were that there are 4 main lineages of the Chytrid BD fungus, 3 of which were found globally, and only 1 was found only in Korea, and then only on the native frogs in that region. They were not found, or it was not found on any invasive species there. And so that gave rise to the Korean Peninsula origin theory. This is still just an idea. It is backed by some evidence, but it is not the end-all say-all. So the results from that 234 sample study indicated that the BD Asia 1 strand, or lineage, is the ancestor of the BD GPL lineage. And I'll show you an infographic in the next slide that kind of explains that in a better sense. And so this also concluded that the spread of the disease did not happen thousands of years ago, which was previously thought. It actually happened between 50 and 120 years ago. And then this kind of coincides with rapid global expansion of the intercontinental trade. So more people are moving around that fungus, moving around those species, and then it kind of just exploded. So this is a genetic diversity and phylogenetic tree of those 234 samples. So we're going to start looking at that outermost ring, this blue ring, that coincides with the year that that sample was collected. And then the inner ring to that coincides right here with chytridiomycosis. And so Chytrid BD can cause the disease called chytridiomycosis, and this is just showing if that sample tested positive, negative, or if the results were inconclusive. The innermost ring to that rainbow colored one, coincides with the continent. And so this was the global collaboration, and so there is a wide variety of continents represented here. And then that innermost ring is coincides with the lineage of the Chytrid BD fungus. And so I had said that BD Asia, which is this blue region here, that is the ancestor to this green region, BD GPL. So as you can see here, there are the four main lineages. This black area represents some hybrids that they're not 100% sure where they go, or this could be a whole new kind of branch of a lineage. It's not conclusive yet. And so going back, scientists went to museum samples. They wanted to see if there were any other preserved specimens that tested positive for this. And so the African clawed frog was actually the first frog that they have concluded tested positive for this. So this was back in 1933. That is when that sample was preserved. And so what they did is they went back and did a skin swab to test if those zoospores were still present in that animal. And so this isn't super surprising because the African clawed frog got shipped all over the world to labs everywhere, and this was used in developmental biology and specifically pregnancy testing studies. And so it's not surprising that these frogs could test positive for it. And then moving on to the spread, so there's a lot of conclusive evidence that humans contributed greatly to the spread of this disease through the international pet trade, so harvesting wild animals and shipping them around the world without any testing. So people could unknowingly be spreading this all over. Research animals like that African clawed frog, that is not the only amphibian that has been used in laboratory testing. So there are plenty more, and back in 1933, or a little while after that even, there was no good way to get rid of lab animals. So sometimes scientists would just release them into the wild, which wreaked havoc on ecosystems, but also spread that disease. And then, in addition, with intercontinental trade, there are always stowaway animals on ships or planes or cars or anything like that, and that is just another mode of moving stuff around. And in addition, a natural way that it can spread through any water system, but there is some evidence that it can be transported through rain, wind, and storms. So those zoospores are so small that they could be contained in water droplets that become evaporated and then disperse kind of in a natural way. And so this is just a brief timeline looking at the first described positive specimen there, and then looking at where and kind of when it was distributed around the world. It's not super important to know dates, but it just kind of shows the progression through the world. And so what is chytridiomycosis? It is a potentially lethal skin disease of amphibians, and so far, as of 2018, it has been known to infect 501 frog and salamander species, and it can cause mass mortalities. So this is a quote from National Geographic. They said, "A team of 41 scientists has announced that the pathogen, which humans unwittingly spread around the world, has damaged global biodiversity more than any other disease ever recorded." And so, who gets infected? So it's many frog species, some salamander species, but they get infected by a different species, kind of. So it is BD sal, and that sal represents salamander. And then there was a single Sicilian species that tested positive, but that was in captivity, and they think it was an accidental cross-contamination by the zookeeper. And then the hardest hit genus is Atopolis. And so if we look over here, you can see that in this image. If we look up at the key, these three last colors represent a 90-degree or higher severity of decline of population. And so you can see here, those reds and oranges show that this genus is not doing well among this Chytrid BD, chytridiomycosis disease. And so over here, it says 71 of the 113 species within that genus are now presumed extinct. And that is awful. That is a large majority of that genus is now extinct, most likely due to this fungus. So of the chytrid-stricken species, 90 have gone extinct or are presumed extinct in the wild, while another 124 have declined in number by more than 90%. And so we're going to talk a little bit about how Chytrid BD and chytridiomycosis affects different life stages of a frog. So just a reminder, frogs start out as an egg. They lay eggs, they grow into a tadpole, they become a froglet, and then they become an adult frog once they lose their tail here. So as a tadpole, because BD is keratinophilic, which means keratin loving, and in this sense that means that the Chytrid BD goes after the keratinized parts of animals. And so in tadpoles, those keratinized parts are the mouth parts. Those are the only parts of the tadpole that have keratin in it. And so this can cause mouth deformations that I'll show in the next slide. But because it is such a small part of the tadpole that is infected, they act as carriers of BD in their environment. And that allows Chytrid BD to reproduce and kind of maintain that in the environment. So these are two images of deformities. This first one, A, is a normal, healthy tadpole. So you can see the jaw sheath here and the tooth rows there and there. In B, this shows depigmentation, so it is white because there's no pigment. And here there's also depigmentation, and then the tooth rows are deformed and also depigmented. Moving into the froglet stage, so tadpoles metamorphosize into a froglet. This is the highest mortality of any of the life stages. So those deformed mouth parts actually result in a loss of mouth parts in froglets. And in addition, the outer external skin becomes more keratinized, so there's more keratin in it, which means there's more of an area for BD to make its way into the animal. And so this typically happens on the underside, so the belly side of a frog, specifically focused on the drink patch area here, the thighs, and then the digits. So the digits are the fingers and the toes of that froglet. And so this is just one more image showing on a frog or a froglet. The belly, the thighs, and the digits are where the keratin is, and on a tadpole, it is the mouth parts. So it's the red coloration in this image shows where keratin is, and therefore that is where Chytrid BD infects them. As an adult, it's pretty much the same as a froglet, but more behavioral changes happen. So they kind of stop eating. That is anorexia. Because they stopped eating, they don't have energy, lethargy, low energy levels, and that can lead to abnormal posture and behavior, and then also hyperkeratosis, which is the thickening of that outermost keratinized layer of the skin. This can actually be 2 times thicker, all the way up to 30 times thicker than normal. And then because those zoospores are in their skin, their immune system wants to get rid of them, so they actually start to shed their skin. And so this is an example of hyperkeratosis. You can see in this first image discoloration. So it's supposed to look similar to the second frog. These are the same species. So this is discoloration, and then these white dots are lesions. So that is caused by that fungus. In the second picture, the coloration is better, but there's shedding on the toes. And then in this third picture here, there's discoloration on both frogs, but then there's also a little bit of skin shedding there and there. So overall, the infection intensity is key. So frogs have about a lethal threshold of around 10,000 zoospores. At 10,000 or above, this impairs the electrolyte transport and cardiac arrest occurs, so a heart attack occurs. And then some species of amphibians actually don't have lungs, and they breathe completely through their skin. And if hyperkeratosis goes all the way up to 30 times thicker than normal, the frogs or the amphibians cannot breathe efficiently, and this actually leads to suffocation. And so this is just a microscopic view of what you could see if you were looking at a lethal infection of, say, a frog. So these white circles all over up here, those are thallus, or thalli, multiple. So remembering back to the life cycle, that was stage 2 of the Chytrid BD life cycle. So they are all up here. They are going to develop into zoosporangium and release more zoospores into the environment. This is a lesion up here and a lesion over here. So those are like those white spots we saw in the previous picture. And then this is overall just severe hyperkeratosis, so that thickening of the skin. And so scientists use skin swabs to detect this. This is done with a cotton swab-like instrument, and they do about 30 strokes on every individual that they test, focusing on the belly, specifically the drink patch area, both thighs, and then the back toes. So they do about five strokes on each of those locations, and then they run a qPCR. And so they amplify the protein that they find, and then with that they are able to calculate about how many zoospores that individual has. And with that information, they are able to make graphs like this and charts that look at different ecotypes, look at how infected they are. They can compare it by species. Here they can look at different locations and look at the zoospore thresholds. They can do all sorts of statistics with that information. And so current research that's being done, Nature published Saved by a Vaccine. This is a really cool discovery, but not super applicable to wild animals. This is good for pets and captive animals, such as in the zoos or something like that. Then these next two, there's research being done that evolution is kind of happening in front of us. So some frogs can survive this, and then if they go on to reproduce, their offspring have a good chance of also surviving it. So they can kind of outbreed the fungus, and eventually it won't have a negative impact on them anymore. This next one looks at eDNA, which stands for environmental DNA, and so this is scientists going into environments, taking samples of soil, of water, off of rocks, off of trees, stuff like that, to see if Chytrid BD is there yet and kind of how bad it is. So this can give them an early cue if they can detect Chytrid BD before it gets out of hand. This one is kind of cool because it's looking at microfauna. So these guys are little Daphnia, also known as water fleas, and they can actually eat those zoospores, which can be a natural way of reducing that in the environment. And then a cure for Chytrid, they created an antifungal that they can spray on the environment, which will kill that fungus. It's cool. It's not super realistic to be able to just go and spray environments all over the world. It's also quite expensive at this point. And so just to recap, amphibian declines are happening all over the world. We are losing them at a rapid rate. Chytrid BD is a fungus that causes chytridiomycosis, which affects the keratinized layer of frogs and amphibians in general. It has different impacts at different life stages of the animal. Current testing uses a cotton-like swab and skin swipes to run a qPCR and then to figure out how many zoospores are present and collect data that way. And current research is kind of all over the place, but they have some really cool ideas, and hopefully soon they will have a good idea on how to kind of eradicate it, or at least control it in the natural environment. Thank you for watching, and I look forward to reading your comments and questions below. And then here's just a short list of my works cited.